15.4, we're going to be looking at alternations of chromosome number, so ploidy, and also some genetic disorders. Some of this will be review, non-disjunction. I know we spoke about that when we were looking at karyotypes. Recall that during meiosis 1, homologous chromosomes separate. So this is meiosis 1. We can see that these homologs are being separated. This one's going to this side, this one's going to this side, so that each cell produced from meiosis 1 gets a copy of that homolog. Well, non-disjunction of the homologous chromosomes in meiosis 1 will result in these two chromosomes sticking together, and they'll go to the same cell, meaning that this cell gets an extra copy of that chromosome, and this cell does not get a copy at all. Now in the image on the left here, as a result of this, we can see gametes getting an abnormal number of chromosomes. We started with 2 pairs of homologs, so each of these cells down here should have 2 chromosomes. However, since n would equal 2, since there's 2 homologs, we have a cell that's said to be n plus 1, another gamete that's also n plus 1, and then 2 gametes that are n minus that one missing chromosome. These cells, these gametes that have an abnormal number of chromosomes, are aneuploids or have aneuploidy. And we can also see here where these two terms would come into play, monosomic and trisomic, mono and tri. Now if one of these two cells happen to be the gamete used in fertilization, then the zygote will have, of this short chromosome here, will have the appropriate number of chromosomes for that one. It'll have 2, one from this gamete and one from the other gamete. But it would have 3 copies of this particular chromosome, 2 from this gamete and 1 from the other gamete used in fertilization. We had previously used trisomy 21 as an example of this process, which is Down syndrome, where a zygote will happen to have 3 copies of the 21st chromosome. We can also see that non-disjunction occurs in meiosis 2, and that would be when sister chromatids fail to separate, resulting in a cell that has 2 copies of a particular chromosome. So non-disjunction can occur both in meiosis 1 and in meiosis 2, but what happens during this non-disjunction is a little bit different than this non-disjunction. In meiosis 1, it's the sister or the homologous chromosomes not separating, and in meiosis 2, if it happens, it's because the sister chromatids aren't separating. Next, we're going to be talking about polyploidy here, which would be many sets of chromosomes. Triploidy, right here, and tetraploidy are rare in zygote cells of humans, but they could happen. A triploid zygote could occur if an abnormal diploid egg existed as a result of total non-disjunction and happened to also be fertilized by a haploid sperm. Now, by total non-disjunction for this 3N state in humans, I would be talking like in meiosis 1 there. This happened to every single one of the 23 pairs of chromosomes. Or if it were in meiosis 2, this were to happen to all 23 pairs of the sister chromatids. That is one way humans could be triploid. A tetraploid zygote could exist if it failed to divide after replicating its chromosomes. So we get a copy of the chromosomes from mom, we get a copy of the chromosomes from dad, so that's 2N, and then keep in mind that that zygote then goes through the cell cycle. Well, it would make a copy of those chromosomes in the S phase, and we could get a 4N zygote if basically cytokinesis never ever happened, then there would be 4 copies of each of those chromosomes as a result of the S phase going through the DNA replication. Some species, namely plants, they do exist in these polyploid states naturally. For example, bananas they exist as triploid. Wheat happens to be hexaploid, and strawberries are octoploid. While polyploid plants are not uncommon by any means, animal species are. There are a few exceptions to that, of course. There are some species of fish and some species of amphibians that can exist in a polyploid state. Next, we're going to look at different ways that chromosomes can change structure. We're going to be looking at 4 deletions, duplications, inversions, and translocations. And before we do, basically this side here on the left represents the before, and the side on the right represents the after. It's quite straightforward. A deletion removes a chromosomal segment. So we can see right here in the before we have A, B, C, D, E. We have a section of nucleotides right here in D, and over here D has been completely deleted. So the way that this chromosome will now function, operate, specifically when it's used to make proteins, it's going to be different. Duplication, here's our before, we have A, B, C, D, E, and after duplication, A, B, C, B, C, D, E, we can see that a section of DNA has duplicated itself, and now we'll have 2 sections of the same genetic information, which obviously could affect potential proteins made from these genes if this section happened to be a coding region of DNA. Keep in mind that there are some sections of DNA that are not used in a gene, so this potentially could be okay. Same with the deletion, provided that they do not occur within a functional unit of DNA. An inversion, we have a before and after. We have 2 sections of DNA kind of switch or flip-flop positions, so that A, B, C, D, E would be read A, D, C, B, E. So we had our D section and our B section kind of switch. That is an inversion. And the last one that we're going to look at is translocation, which we had spoke of or I mentioned previously when we were talking about leukemia, which is where one region of a chromosome and one region of a second totally different chromosome exchange regions of DNA. So the M, N, and O are going to plop onto this chromosome, and the AB region is going to plop onto this chromosome. So now we have, instead of a change going on within one chromosome, we have a change going on within 2 different chromosomes. In the last section, 15.5, some inheritance patterns are exceptions to standard Mendelian inheritance. So our next slide, we're going to be looking at imprinting, which for anyone that's been following Twilight, you might understand why I have this picture here. And if not, you have a little bit of homework, and that's have a huge marathon with those fantastic movies. So imprinting, it is real. It can actually happen. Not exactly the way that we saw it going on with Jacob here and a little infant child. This actually happens at the genomic level, so it's called genomic imprinting. So what is genomic imprinting? Well, genomic imprinting involves a dependency on which parent, mom or dad, that passed along a specific allele for a trait. Geneticists have identified roughly 36 traits that function in this manner. So what does that mean? Essentially, you get allele A from mom, you get allele A from dad, and one of those 2 alleles is silenced, meaning that the other one is expressed. So for a certain gene, only one allele will be used, and it depends on if that allele came from mom or dad. It could alter the function of that gene. Anyhow, scientists have identified about 3 dozen genes or traits that function based off of genomic imprinting. This occurs during gamete formation, and as we had just mentioned, it results in an allele becoming silenced for specific genes. As a result, the zygote, after fertilization, will express only one copy of the gene since the second had been silenced. Our next slide will show us an example using the IGF2 allele, which is a gene in mice that determines how large or small a mouse is. So here we are looking at mice. There's normal mouse size, or the wild type, and then there's dwarf mice. IGF2, better known as insulin growth factor 2, was one of the first imprinted genes that have been discovered. In this particular gene, only the paternal chromosome is expressed, and the maternal gene is always inactivated. So here is a mouse. This happens to be, well, I guess it doesn't matter if it's male or female, because this mouse will get a chromosome from its dad, and it will get a chromosome from its mom, and the IGF2 allele from this mouse's mother will always be silenced, and the one it gets from dad will always be expressed. So, how could we get a dwarf mouse then? Well, it would ultimately be determined by the dad mouse, because the mom's chromosome would always be silent, so it would be based on the dad. If this happened to be a female, her child, it wouldn't matter if she passed on the mutant allele or the normal allele. If this were mom, either way, that allele would be silenced in the child, because once again the maternal IGF2 allele is silenced. However, if this were a male mouse and he happened to be heterozygous for this particular gene, if this guy passed this chromosome on to his child would be fine. If he passed this chromosome on to his child, then the child would be a dwarf. Now, what's interesting is if this happened to be a male mouse, this was the chromosome he got from his mother. However, during gamete formation, this inactivated allele, it resumes functionality, especially if it gets passed down to its child. However, this would not affect the size of this mouse. It's always this one. So that would be how this particular allele could be passed from parent to offspring, and it would be completely determined based off of the paternal chromosome that this guy passes down. I guess another way to think about that is once this guy inherits both of those chromosomes, they're both his. So which one does he pass down? This one or this one? One last note on genomic imprinting. It's not always the dad's chromosome. It's just dad's chromosome in the instance of the IGF2 allele. And the last little bit we're going to be looking at is organelle genes, which are genes that are found outside of the nucleus, and they're found in organelles within the cytoplasm. Hopefully you recall that there's 2 organelles that do have their own DNA, and that would be the mitochondria and the chloroplasts, which is why we actually think these were once organisms that existed outside of cells, and that's because they have their own DNA. Now with these 2 particular genes, we're going to be focusing more or less on plants, because plants have both chloroplasts and mitochondria, and the expression of those particular genes will depend entirely on the maternal parent, because the zygote's cytoplasm comes from the egg, meaning that all of the chloroplasts and mitochondria in plant zygotes come from the egg. The pollen contains none of those organelles. The pollen just contains the chromosomes, no cytoplasm. The egg has all the cytoplasm with those organelles.